Hello student. In today's lecture we are going to talk about the ultrastructure of bacteria. Now in last few lectures we have seen that on earth there will be two different types of bacteria or two different types of organisms are available, and these two types of organisms are nothing but the unicellular and multicellular organisms. This classification of organism is on the basis of type of cells, number of cells. Say for example, when we talk about the multicellular organism, large number of cells gathered together or organized together, and this organization leads to form the organs. So cells unite with each other, they perform a particular function, and that we called it as a organ. Now these organs have a particular type of function. When these organs combines together, we called it as an organism. So in a multicellular organism, large number of cells are gathered together, or we can say they combine with each other and they perform a particular task. But in unicellular organism, we know very well that a single cell is there, and this single cell perform the different types of tasks. So depending on the type of cells, whether multicellular or unicellular, we can say there will be two categories of the cells or two categories of the organism. One we called it as a prokaryotic organism, and another we called it as a eukaryotic organism. So in last lecture we have gone through the prokaryotic structure. Prokaryotic structure include the or prokaryotic organisms include the bacteria. So in today's lecture we are going to concentrate on the structure of bacteria. Now we know very well that the bacteria is the unicellular organism, and this unicellular organism is having the particular structure. They have a different functions. It is performed by the single cell or unicell. Now each cell or bacterial cell is having the external cell wall. To inside this external cell wall there is a presence of cell membrane. Locomotory organ like a flagella is also present in some bacteria. Then in some bacteria there will be a presence of inclusion bodies, ribosomes, genetic material, and the most important thing which is present in the bacterial cell is a plasmid. Now cell wall, we call it as an external layer, so it is present towards the external side of the wall or external side of the bacterial cell, and that's why it is known as the cell wall. This cell wall acts as a protective material or protecting material for the bacteria. Followed by the cell wall, internal side to the cell wall there is a presence of cell membrane, and in some bacteria there will be a presence of flagella which we call a locomotory organ. So for locomotion from one place to another place, flagella is present. Now in some organism flagella is single, or in some biflagellated structure is there. So depending on the number of flagella there will be a different we can say the bacterial cell is there. Inclusion body is nothing but the material which we called stored material or stored food material in a cell is also present. Now ribosomes, then genetic material and plasmid, these are the most important organelles, or we can say most important structure present in a bacterial cell. Plasmid particularly is the most important, and it is only present in a bacterial cell. That is the extra genetic material in which is present in a bacterial cell. Not all bacterial cells shows the presence of plasmid, whereas some of the bacteria shows the presence of this plasmid. Now here you can see the typical bacterial cell structure is given. Here on the external side you can see that label is given, that is known as capsule. Capsule is an extra protective covering, or it is a slime layer which is present external to the cell wall. So this capsule or slime layer is present in some and absent in some also. So we can say it is the extra protective covering present to the cell wall or in a some bacterial cell. This capsule is followed by cell wall. So cell wall in a bacterial cell is somewhat thick in size. It is protective in nature, and followed by this cell wall you can see there is a presence of plasma membrane. So here the green color structure you can observe that we called it as a plasma membrane. This plasma membrane is followed by the presence of cytoplasm. In cytoplasm we can see there is a presence of mycosomes, plasmid, ribosomes, and along with that you can see again here the presence of pili. Pili is the most important structure which is present in bacteria. It is not compulsory in all bacteria, it is present in some bacteria, whereas it is absent in some bacteria. So this pili and flagella is having a specific function which we are going to discuss in upcoming lecture. So pili and flagellum, these are the two structure which is present in the bacteria. This pili and flagellum is an additional structure, it is not compulsorily present in all bacteria. So this is nothing but the typical bacterial cell. Now when we talk about the cell wall, the cell envelope is composed of plasma membrane and cell wall. So cell wall is the external covering. It is as in another organism or other organism, the bacterial cell wall provides structural integrity of the cell. So it is the only single cell, and that's why it is the structural integrity which is given by the single cell. Now in prokaryotes or prokaryotic cell, the primary function of the cell wall is to protect the cell from the internal turgor pressure. So cell wall is a protective in nature. It will maintain the pressure which is present from the inside, and it also protects the cell from the external pressure also. So it is nothing but the protective in nature. Now this cell wall differs from that of all other organisms by the presence of peptidoglycan, which is located immediately outside the cytoplasmic membrane. So we can say that in eukaryotic cell and in prokaryotic cell, this is the difference, that the peptidoglycan layer is present in a prokaryotic cell, particularly in a bacteria, and that is peptidoglycan provides extra rigidity to the cell wall. Peptidoglycan is made up of polysaccharides backbone. So it is nothing but the chain of polysaccharide. It is a chain of sugar, and this chain of sugar act as a backbone for the cell, consisting of alternating N-acetylmuramic acid and N-acetylglucosamine residues in equal amount. So in simple way we called it as a NAM, that is N-acetylglucosamine or muramic acid, and N-acetylglucosamine. NAM and NAG, these are the two different structure or residue we can say. These two residues together produce the structure that we called it as a peptidoglycan layer. So here you can see the peptidoglycan layer is there, and the chain of side chain of peptide is also present. So here you can see the NAM and NAG is N-acetylglucosamine and N-acetylmuramic acid. They are present in a alternative structure, and along with this you can see there is a presence of cross bridge of amino acid and side chain of amino acid. So in this way we can say that this particular structure get intermingled with each other, and they provides a rigidity to the cell wall. Now here you can see this is nothing but the N-acetylglucosamine which is present over here, and side by side you can see there is a presence of N-acetylmuramic acid. So these are nothing but the N-acetylmuramic acid with attached peptide bonds, or having the peptide bonds with the side chain of amino acid. Now this is nothing but the structure of the bacterial cell wall, which is provided with the additional material. Now here you can see this is nothing but the amino acids, It is the side chain of amino acid which is attached with the NAM, and these are nothing but the cross bridge of amino acid. So in this way, when we talk about this particular structure, peptidoglycan layer, you can see it is a gram positive bacteria, and in a gram positive bacteria we can say peptidoglycan layer is shown like this, and that's why the cell wall of this particular bacteria, that is gram positive bacteria, is having somewhat thick structure. So here you can see the membrane is there, that is cytoplasmic membrane is there, and outer side to this cytoplasmic membrane you can see the peptidoglycan layer is present, which is in a thick size. Whereas when we talk about the gram negative bacteria, the peptidoglycan layer is very small and it not provide that much support, or we can say it is not that much thick as it is in a gram positive bacteria. So membrane is there, this membrane is followed by peptidoglycan layer, and after the peptidoglycan layer, lipopolysaccharide and protein structure is also present on the outer side of the gram negative bacteria. So this is the basic cell wall structure which is having the difference in gram positive and gram negative bacteria. Now, as earlier we discussed that there will be two different types of bacteria available on the earth, or categorized in a two different way. We call them as a gram positive bacteria, and in another way we called it as a gram negative bacteria. So these two bacteria is having the difference in their peptidoglycan layer, and that's why we can say they shows the different structures. Now followed by the cell wall, there is a present cell membrane. So it is the internal material or internal cover. This is made up of phospholipids and protein and carbohydrates, forming a fluid mosaic. So in first year we have discussed this fluid mosaic structure. So this fluid mosaic structure, in which the phospholipid is present, in which the protein is present, and along with that carbohydrate is also present. So this is nothing but the structure which is present in cell membrane. Now this cell membrane surrounds the bacteria, and it is the most important organelle because it also helps in the protection, it also helps in the size and shape of that particular bacteria. It controls the movement of substance in and out of the cell. So the material which is present from the outside will get entry towards the inside only through this cell membrane, or it is also restricted by this particular cell membrane. So this is nothing but the cell membrane. Now here you can see the cell membrane is shown over here. So this type of fluid mosaic bilayer structure is present in a bacteria. So this is nothing but the phospholipid structure, and this is the scanning electron micrograph of the bacteria, which shows the presence of outer membrane, which shows the presence of peptidoglycan layer, and it is also having the structure like phospholipid bilayer of plasma membrane. So this is the scanning electron micrograph or microphotograph, we can say, of the bacterial cell. So we stop with this. In upcoming lecture we are going to discuss about the capsule, slime layer and S layer. What are the difference between these things we are going to discuss in upcoming lecture. Till now you can watch this video 2 times to understand the various concepts. Thank you.